All right. Well, what's in the extracellular matrix? You see an idea of what it's used for. We see its evolutionary implication. Now, what's the composition? Well, once again, for the most part, not counting the mineralized ones, for the most part, we're dealing with secreted proteins and polysaccharides. So let's first look at the polysaccharides. There's a lot of different kinds of polysaccharides that go in the extracellular matrix. We've already looked. Chitin, cellulose. Okay, we see those plant cell walls, fungal cell walls, arthropod exoskeletons. But let's look and focus the ones we find in most animals. We have a number of different kinds of polysaccharides, but most of them fall in the category of, that goes into a jaw-breaking name of this, glycosaminoglycans. Or GAGs for short, because I guess trying to pronounce that will make you gag. All right. These are very common in what we call connective tissue. Now, what are these things? These are a type of polysaccharide. The name glycan, that refers to sugar. Glyco, sugar. Amino, what's that there? Well, what it is these are generally an alternating 6-carbon sugar followed by an amino sugar. Remember, amino sugars have nitrogen-containing amino groups, and you repeat this hundreds or thousands of times over. A sugar followed by amino sugar, another amino sugar, and so on. So many of these things are several hundred to several thousand sugar and amino subunits, amino sugar subunits long. They form very long molecules. Now, one thing about these guys. One, these guys, the polysaccharides. Polysaccharides are very polar. Not only that, many of them have covalently attached sulfate groups. So we see they're sulfated. So that means they have covalently attached SO4 2- sulfate ions on it. So not only are the guys superpolar, they're charged as well. Now, a common one of these, and I have first-hand experience with it from my last hospital trip, this stuff called heparin, it's a blood thinner, it's one of these. It inhibits blood clotting, which is good if you have a clot in your lungs. Okay, now, so a lot of these things are sulfate. Now, what happens? They're extremely polar molecules. They're too large to kind of like, quote, dissolve in water, but when you put them in water, they attract and stick water molecules all around them. So what happens is they soak water up like a sponge. So if you take a couple grams of that stuff, dry powder, throw it into a glass of water, let it sit for a while, you turn that glass of water into some kind of jelly-like substance, because it sucks that much water up. Likewise, if you have a lot of it in some kind of connective tissue, you get something that's jelly-like, rubbery-like, or whatever. Think of cartilage, right? You ever bite a piece of gristle? We go down to a cheap steakhouse or something, like rice. And then you get that gristle-rich, cheap steak, and you're constantly chewing on gristle. Okay, you know it's got that rubbery feel. You can thank, at least in part, these glycosaminoglycans for that stuff. So that's one kind of thing. Now, many of these form even larger structures called proteoglycans. With a name like that, suggests that there's a protein involved. And sure enough, it is. What a proteoglycan is you have this long protein, sometimes called a core protein. There's another name for it, I forgot, syndecan or something. But anyway, it's a long protein. This protein is almost all alpha helix, so it's an extremely elongated molecule. And covalently attached in numerous places to this core protein here are going to be lots of glycosaminoglycans. Hundreds of them. So you have the core protein with scores to hundreds of glycosaminoglycan chains coming off of it. These things with molecular weights in the millions are so large that you can see them easily under an electron microscope. They look like a molecular bottle brush. These things can be micrometers long. They're huge. And once again, with all those very polar glycosaminoglycans there, they soak up water like a sponge. So individual glycosaminoglycans and proteoglycans, those are major components of a lot of our connective tissue. So that's one thing. And all these different polysaccharides. So these are some of the major types of polysaccharides. Okay, questions on that? Well, polysaccharides are only part of the story, so let's take a look at extracellular matrix proteins. The majority of extracellular matrix proteins are glycoproteins. Glycoproteins, of course, are proteins that have covalently attached complex polysaccharides. And there's a number of different families. We're going to look at a couple of the major and most important families, but there are many others as well. Let's look at a couple major ones. Perhaps the most abundant and most common family of proteins you've already heard of. The collagens. Many times you people call it collagen, but it's actually a whole bunch of related molecules. You sometimes see in cosmetic ads and stuff, oh, this stuff has collagen in it for smooth skin and stuff. And if you believe that, I got a bridge in New York City to sell you. It's probably flooded right now. Because, hey, look, one, it's coming from animals. It's not going to do you any good. It's coming from cattle and stuff. It could even possibly get an allergic reaction. And two, it's not going to get through your skin because it's a huge molecule and won't penetrate through your skin. But you can sell lots and make lots of money that way. Okay, so yes, it's my collagens. Okay, who cares? All right, well, collagens are actually a big family of molecules. And what happens is you have these individual proteins. And there's about 30 different genes for them, and they're called the tropocollagens. These are subunits of collagens. And their molecules about 60,000 molecular weight. One thing about them is they're almost all alpha helix. So they're very elongated, thin molecules. So now what happens? You get the tropocollagen. Okay. Now, there's different types of tropocollagens that are put in certain categories. And there are certain combinations that can be allowed. But basically, to make a collagen, you have to take 3 tropocollagens, and they line up kind of like this to form a triple coil, or a triple helix. Each one of these lines represents a tropocollagen. Now, you cannot make any combination of tropocollagens and put them together. There are certain rules of the game, but still, because there's about 30 different tropocollagen genes in vertebrates, you can make hundreds, if not thousands, of different types of collagens. But that's our basic structure here. Now, some of these guys line up end to end to form fibers. You can find lots of these fiber-forming collagens in things like ligaments and tendons. Other kinds line up sort of like so to form thin sheets. Think of those thin sheets of connective tissue that line our pericardial cavity, our pleural cavity, our peritoneal cavity. Think of those. Those are mostly the sheet-forming collagens. So there's a whole bunch of them. Another thing about collagens is they have a very unusual amino acid sequence. Almost all of them consist of the amino acids glycine, followed by proline, followed by something else, we'll call it X, repeated over and over and over again. So most of the collagens consists of 2 amino acids, glycine and proline, and then you have any of a number of others. So that's kind of very unusual structure. Now these are both quite polar, these are both pretty much polar amino acids, so collagens, not surprisingly, are polar molecules. With the covalently attached polysaccharides, it makes them even more polar. Now, aside from cosmetics, I bet you ran into collagens and didn't even know it. So we're going to do a little nostalgia trip here. Go back to your childhood, right? Mommy, I want a snack. And Mommy goes out to the cupboard and pulls out this little small box of something. Jello, brand Jello. And she pours a little box with 100 grams of the stuff in there. Pours it into a couple liters of warm water and sticks in the fridge. And a few hours later, it's Jello. Okay, what is in that little box? 100 grams. Most of it is sugar, unless you get the sugar-free version. The boxes are even smaller. And then you've got artificial color, artificial flavor, but the main ingredient in there is collagens. Denatured collagens, in fact, because they obtain the stuff by getting horse and cattle tendons, hooves, and things like that, and they boil it. Boiling proteins denatures it, but they extract the stuff out, and then they dry it up, and then they put it full of sugar, artificial color, artificial flavor, and package it and say, "Here, kiddies, have some horse tendons. Good, love it." Yep, that's what you're eating, is horse tendons and hooves and cow tendons and hooves. Now, if you haven't bothered to read the label on the side, even though collagens are proteins, you look at it and nutritionally, it ranks as 0 for nutritional protein. Reason why is this? It's made mostly of glycine and proline, and guess what? We synthesize glycine and proline. They are not essential amino acids. We make the stuff from scratch. And too little X around to count for much. Most of those are not essential amino acids. So even though collagens are proteins, nutritionally they rank as 0 because it's almost all, it has almost no essential amino acids that you have to take in from your diet. So legally they cannot consider it as a source of dietary protein. So, now, making Jello gives you something. Picture at least half of that box is sugar and color and flavor, and the other half or less is the collagen. So you get like maybe 50 grams of collagen, dry collagen. You put in a couple liters of water, and it absorbs water so much that it forms a gel. And it will work with other polar molecules too. Case in point. Substitute the water with a 50% ethanol solution. Yes. Vodka. Or whatever. Pour it in, and lo and behold, it will still gel. And this is the jello you do not give to the little kitties. This is the jello that will put you butt on down hard and fast. And being a future scientist, was I in grad school, right? I played around with some of my friends, had some grain alcohol. So we're going to see how strong of a Jello shot can you make. Up to 70% ethanol, and it will still gel. And boy, let me tell you, oh man, you have to be real careful with those. Okay, but yeah, because here again, ethanol is not as polar as water, but collagen will be able to bind in sufficient amounts to generate even fairly high concentrations of ethanol, a gel. And there's a certain point where it won't work and you get this goopy stuff. We found that out in experiments. But you can see it's very polar. And of course, collagen in connective tissue does the same thing. It soaks up water like a sponge, so it also contributes to the gel-like, dense gel nature of connective tissue. So collagens are one important family. They're probably the most abundant of the extracellular matrix proteins. And different collagens have different cellular functions and organismal functions. We'll see later on what happens when you end up missing certain types of collagens. It is not a pretty picture. Well, that's one family of glycoproteins. Another one that are quite abundant are called the fibronectins. They're probably the second most abundant family. These are very large molecules. They frequently bind to collagens. Usually it's two large molecules joined together, two subunits. Another one that you find called the laminins. Form what's known as our major components of what's called the basement membranes, or the Latin term for all you medical people is basal lamina, that holds various kinds of endothelial cells together. Think of your gut lining. What you got in the lining of your gut is you have a whole bunch, like your intestines for instance, you have a whole bunch of these sort of column-shaped cells. And these are the ones that secrete the enzymes and pick up the nutrients. Now, you want these guys to hold together, so what they have is this thin layer of extracellular matrix, the basement membrane that glues these cells together to form a tissue. If you suddenly made that disappear, your intestinal lining would disintegrate. That would be very bad, right? Okay. The basal laminas, or basement membranes, are loaded up with these giant glycoproteins, the laminins. One other one I'd like to mention, because they're cool, and they're important too. What are called the elastins. Elastins form what we call elastic or stretchy fibers that we find in a lot of extracellular matrix. What these things do is give extracellular matrix a little bit of elasticity, a little bit of stretchiness. Because elastins themselves, you can take one of those guys and stretch it to 7 times its normal length, let it go, and it would snap back. Like a rubber band, but even more so. As a matter of fact, the way it works is very much like the latex protein in rubber. It's actually very cool. Now, think of what would happen if your connective tissue did not have a little bit of elastins in it. Has anybody here ever torn a ligament? That was a lot of fun, wasn't it? It's double over and practically sob in agony, right? It's incredibly painful. The reason only one person in this class has torn a ligament. Oh, you did too? Oh, you did too? Okay, 2 people in the class have torn ligaments and would not want to repeat that experience, is our ligaments have quite a few amounts of elastins. So they can stretch, obviously there's limits to that, right? We found out the hard way. But they can stretch to a certain extent and not tear thanks to the elastins. If it wasn't for the elastins, we'd be tearing our ligaments all the time. That would not be good. Most of the time when you tear ligaments, either bad fall or sports, right? Sports in your case? Okay. Bad fall. Okay. Yeah. All right. Oh, that'll do it. Oh, that'll do it. Okay. So elastins are very cool. And here again, a lot of our connective tissue has varying amounts of elastins in them to give them a little bit of stretchiness so they don't rip. They stretch a little bit. Now the cool thing about elastins is this. The nature of why are they stretchy? How do you get a stretchy protein? Well, this and the latex protein of rubber that makes natural rubber have the same basic principles. It's actually quite cool. You have a very elongated molecule that's a lot of alpha helix. And then you have these regions that fold up because they're full of hydrophobic amino acids trying to avoid water. So you have these repeated regions here. They're all folded up. And they're folded up like all proteins fold up to keep the hydrophobic amino acids away from water. Now you stretch these guys. What happens when you stretch them is now you're pulling the hydrophobic regions and stretching them out. Okay, as long as you're applying that mechanical force to them, you overcome that. But when you release that force, these little regions fold back up again. Since they're small, they can actually fold really, really quickly. So what happens is now they try to go back. In effect, when you take a rubber band, at least natural rubber, or an elastic fiber and stretch it, what you are feeling, the resistance you are feeling, is the energy of hydrophobic interactions. You are breaking them. They're weak, but it does require energy. You're breaking these things, and when they try to refold, that's the stretching, that's the pull that you see on it. So in effect, when you stretch a rubber band or something like that, or a latex glove or something, what you are doing is turning hydrophobic interactions, the energy of that, into something you can actually feel and measure. So that's a neat little thing about the elastins. All right. So those are some examples of what goes into the extracellular matrix. Let's see how much time we have. Not much. So what we're going to do, we'll deal with the extracellular matrix signaling on Tuesday. But what we're going to do now